Welcome to SARC Talk. SARC is a nonprofit cancer research organization that develops and manages clinical research trials in pediatric and adult sarcomas. SARC Talk is a podcast to discuss activities of SARC, interview sarcoma research leaders, and review scientific initiatives in the sarcoma field.
Welcome to SARC Talk 18. My name is Scott Okuno. I'm medical oncologist at Mayo Clinic and Chief Medical Officer of SARC. At SARC Talk, we interview interesting people who are connected to sarcoma, where we can share and spread knowledge about sarcoma. All episodes are posted on our website, Spotify, YouTube, SARC channel, or wherever podcasts are available. Please subscribe and send comments because we do look at them. It is my pleasure today to have Dr. Crompton here as our guest on SARC Talk. Dr. Crompton is an associate professor of pediatrics, Harvard Medical School. He did his medical school at Boston University. He did his pediatric residency at the University of California, San Francisco, and came back to Boston to do his fellowship at Boston Children's Hospital at the Dana Farber Cancer Center. He has an active lab, Crompton Lab, and please Google it because he does have a very nice website for that. And his research interests include utilizing proteomics and genomic approaches to expand our knowledge and understanding of cancer biology, identify novel treatment approaches for pediatric tumors and rare sarcomas. Please welcome Dr. Crompton.
Thanks for having me. I appreciate the opportunity to be here.
Thanks for connecting. I have been following. A lot of us have been following a lot of what you've been doing, and you've been involved with a lot of the pediatric realm as well as your publications in the adult realm and some other aspects. So hopefully today we'll get to learn a little bit more about yourself, about your lab, and where you see the vision of the work that you are doing in DNA analysis. So maybe for our audience here, maybe you can share how you decided first to start with become a pediatric hematologist oncologist.
Yeah, well, it's a very academic explanation, to be honest. One thing I always tell patients and families is that I think it's really important that doctors have at least as much maturity as their patients. So pediatrics seemed like a natural fit for me. But I've always loved, sort of mentorship, and I coach my kids' baseball and softball and soccer teams. So I love being around kids. I think when I was thinking about being a doctor and thinking about pediatrics, I did want to go into a field that was specialized, that would give me an opportunity to do both patient care and research, ideally at the highest level, and then also be able to make an impact over the next, whatever years long a career lasts these days. And so there was definitely, I had a short list when I got to residency and to do my general pediatric training. And at UCSF at the time, they were really keen to help us do some rotations in the specialties we were most interested in towards the beginning of our second year of residency. And oncology, hematology, and bone marrow transplant was the first one of the first specialty rotations I did as a resident in training. And I mean, I completely fell in love with the kids and the science. And I had some friends who teased me about how dorky I sounded at the microscope when I was looking at smears with the expert hematologists and oncologists, and I just love the field.
Well, thanks for sharing, and I think we're probably just divine intervention to say that your first rotation was heme, because if you were doing cardiology, you might be in a totally different field. So this is all great for us.
Maybe. I mean, I have a lot of respect for all the other fields for sure. I do think that oncology is this extremely unique space where you can really have longitudinal care of patients who potentially, need a high level of medical care, and where a true focus on academic research is also highly valued because we have so much to learn. It's pretty special in that way. I'm not sure I would have found that perfect Venn diagram in any other specialty. So.
Well, that's true. So mine might not than the sequence, but it's important to know that this is kind of a very unique thing. And then, you look at the whole hemonc space. We focus on a very specific component of hemonc, which is sarcoma, and that's what your lab really focuses on. But a lot of techniques that you'll share with us today are applicable to a lot of different things, and we would just like to hear a little bit more. So, with that, I'd like to have if you share with our group here, how did you get interested in a very specific terminology? And I might use a terminology differently than you might like or what is appropriate, circulating tumor DNA or circulating tumor cells. And maybe you can share with our group, because we have a very broad audience here, of kind of how you got involved with that and how you define those two things.
Yeah, well, I think there is a little bit of serendipity in the world, and I think you always have to sort of be prepared for opportunities. So, I mean, really, the reason I think I have gravitated towards liquid biopsies, sort of that's the umbrella term I like to use the most for the things you've mentioned, um, has components of all that. So when I was training as a postdoc in Kim Stegmeyer's lab, we were focused on a couple different projects in Ewing sarcoma, and one of them was defining the genetic landscape of Ewing sarcoma. So really trying to make sure we understood what all the mutations were that cause Ewing sarcoma tumors to exist, and that's a very most pediatric cancers are rare, but that's one of the more common bone cancers that we see in children. So and so we did that project, and I learned an amazing amount from my mentor, from collaborators at the Broad Institute, computational biologists, and there was also a piece of it that reminded me of my excitement when I was, I think, in high school, when the Human Genome Project started to report out results, and how like mind-blowing that was to be thinking about sequencing genomes, and to be able to do that as a trainee, as a fellow postdoc in a lab, after like having dreamed about that for years as a college student and medical student and resident, was like really spectacular. And then as I was sort of moving towards finishing up my postdoc time and being, starting to think about starting my own research group, and also being recruited as a faculty member to start a laboratory here and at another institution, Luis Diaz came to give a talk, and he was one of the first people to really publish about how you could really detect fragments of DNA that were released from tumors in a blood sample. And he came to give a talk at Dana Farber, invited by someone in our, again, mentors, someone who was a senior person in our pediatric program. And the research blew my mind. I got invited to go to dinner with him as one of the junior folks that took him out to dinner after the talk. And I ended up sitting next to him, and we were chatting for like an hour about how applicable and straightforward it would be to try something like this in Ewing sarcoma. And so I went back to the lab and checked with my mentor. I had some donor-directed funds from a grateful family who still support my lab today, amazingly. And we used that money to try out some things, and it totally worked. And so we were able to detect these fragments of tumor DNA from blood samples we had collected from patients with Ewing sarcoma. And it just felt, on the one hand, like science fiction, like we were doing something, you could see on like Star Trek or something. And on the other hand, I think people probably know when they're doing something that just feels right for who they are, and like the genomics and the math, it like just clicked for me, right? That was what I knew I could be good at. And so when I was starting my lab, we kind of figured this would be like maybe 20%, 50% of what we do. And now, I mean, it's now almost everything we do is focused on liquid biopsy. And part of that's because we really believe it's going to have a big impact on how we care for patients going forward. And also because I think in the pediatric rare cancer sarcoma space, I think we're one of the labs who do it well and think about these cancers that not everybody, and certainly commercial entities, don't spend a lot of time developing tests for. So it's sort of exploded, and now we have more projects that we can handle in this space, and it's nearly 100% of what we do.
Well, thanks for sharing that because it's serendipity is what you said. But, those that are just listening, you should see Dr. Crompton's eyes. They're like almost like, it's like, this is like the best thing in the world. And he used the word geek out or nerd out, but this is really his passion. You can kind of just see it in his face as he's discussing it. And as you said, he went from a 20% component of his lab to almost 100%. And let's just backtrack a little bit because you did say something that I think is important for all of us is that there are a lot of commercial products out there that promote what they would say liquid biopsies. And there are different components, as you said, to that. And maybe you can share like what is the difference between a commercial product versus what your lab is working on, because some of the stuff that we see and we're going to get into are very specific for Ewing sarcoma. Some of the data to validate and how we use it clinically is going to be driven by your lab. How we do it for other sarcomas is going to be driven by your lab, but I'm not sure that's the same thing that commercial groups do. So maybe you can share with our groups how that all pans out.
Yeah, I mean, this is sort of a common theme across different aspects of rare cancer research. I think it's sort of applicable. But just to back up a minute and say that I think there's a couple different ways that liquid biopsies are going to be useful, and I think part of it is understanding better which patients are are most likely to be cured by a lot of our standard therapies, and which ones are at higher risk of eventual relapse and need either more intense therapy or some experimental agents to try to give us a chance to cure patients. And we have not historically had great biomarkers for figuring that out up front or during therapy. Even measuring response early on to therapy has not been so great. And there's a lot of great data out there, really an ocean of data now these days, that liquid biopsies can do that in cancers. But to make that work, you have to have a test that can detect in the blood the hallmarks of the cancer of the patients you're treating, right? And so what that really means is to identify fragments of DNA that are from a cancer cell. You have to differentiate it from fragments of DNA that are coming from other cells, because all of us shed DNA into our blood at very low levels, and only a small fraction of that is from the tumor in patients with cancer. So you have to be able to see the hallmarks, and most people, I think, are aware at this point that what makes a cancer cell different from a regular cell is a mutation, a change in the DNA sequence that all of our cells contain that tell that cell to start dividing, keep dividing, don't stop no matter what the signals are. That's sort of the essence of cancer. And so to find fragments of DNA from a cancer cell, you want to find those mutations, those changes in the sequence of the DNA that's being shed from those cancer cells. And rightly or wrongly, the first tests that were developed, even in the research space, to identify cancer cells, looked for the mutations that are associated with the most common cancers. And that kind of makes sense, of course. But those mutations tend to drive carcinomas, which are totally different than pediatric cancers and sarcomas. And in fact, the mutations we commonly see in carcinomas are quite rare in pediatric cancers and sarcomas. And so as this field was clearly taking off, there seemed to be a need to develop tests that could detect these mutations that were more specific and the common drivers of adult sarcomas, pediatric sarcomas, and pediatric cancers generally. And so that's really what my lab did. And since we had some expertise in doing sequencing and profiling tumor samples, the DNA of tumor samples, identifying mutations from tumor samples of these rare cancers, we were well poised to try to develop some tests that could detect fragments of tumor DNA that were derived from these rare cancers. One thing we don't want to be, my lab doesn't, well, I don't want my lab to be, is a tech-only lab. Like, we're not, there are groups that get really specialized in a certain technology, get really specialized in a certain analytical tool. But we really only want to build something if there's a need. And so there are definitely assays out there that we didn't develop, that we use all the time, that we've just adapted and we profile samples with because someone else invented it. It's a great invention. Don't break it if it, don't fix it if it ain't broke. And we just apply it and use it. And a lot of times we use it in collaboration with the people who invented it. And that's like another amazingly fun aspect of the research we do, is we're just super, super collaborative. And we have a lot of friends in this space and we learn a lot from other people. But there are certain cancer types for which there was no test that was going to really work to detect these circulating tumor fragments in the blood. And so in those areas, we did develop some tools that have been very effective at detecting the presence of cancer and sort of being utilized as a biomarker. A lot of those original tests that were developed in labs for carcinomas have now gone on to become commercially available tests. And what that means is that you can go to your doctor and order the test, and some are covered by insurance, some aren't. But one way or another, those tests can get paid for and returned. That result can be returned to the physician and be put into the medical record, and decisions for care can be made off of those tests. And that's great for patient care. And so whenever there's a commercial test out there that we think is adaptable to patients we care about, we want them to get those tests because those are the most useful tests. They're immediately useful to the patients and their physicians. But now that we've developed some of these research-based tests that seem to be really effective for detecting ctDNA, quantifying the amount of the circulating tumor DNA, and predicting response to therapy, it's now our job actually to get those out into a clinical return of results space. Now, it's unlikely that for rare cancers that are a test that will be used a few hundred times or even just 1,000 times a year is going to be picked up and turned into a financially profitable test. That's what we mean by a commercial test. But there are most academic centers have the ability to take specialized tests, run them in a clinical laboratory that allows for return results. And that's also some of the work we're doing now here in Boston Children's, and Dana Farber is trying to figure out how we can get these tests so that we can return the results to patients, return the results to their physicians, return their results to a clinical trial they may be on, so that those can impact the decision making and eventually improve our care and improve outcomes.
Yeah, and that's, I think for the people that are listening, the component is, yes, do the work, get the science done. Maybe a big company might not take it out because it's not profitable, but what you're trying to do is drive the next steps and improve outcomes. And whether it's through a clinical trial, et cetera, that makes huge sense. And as you said, you're 100% full doing 100% of this work, but the space that your lab is really focused a lot is in Ewing's and rhabdomyosarcoma, but you do have collaborations. And I know you've published work on the circulating tumor or say liquid biopsy space in leiomyosarcoma. And how do people get engaged and connect with you? Because you're going to have an insatiable amount of collaborators, but you only have one lab.
Yeah, well, we've grown a lot, so that's been fun. I think what we try not to say no to good projects. And that's just advice I got. Try not to say no to good projects that I got as a trainee growing up. I mean, we really take advantage of some of the automation and like incredible capacity for sequencing that the Broad Institute provides. So, my primary affiliation is with Dana Farber as my employer, but I also have an associate membership with the Broad Institute. People may not know this, but the Broad did a huge amount of like the Northeast COVID sequencing during the heyday of COVID testing. Their capacity to do sequencing either in a research space or in a clinical return results scenario is like pretty enormous. So we do take advantage of that, and what goes along with all that sequencing capacity is an incredible ability to analyze that really complex, really big data that you get out of a sequencing run, and then all the computationally, all the math geniuses that sit there between the Broad and MIT who can help us analyze the data. So that helps us sort of with our throughput for sure. but what works best is if we partner with a very engaged, committed, enthusiastic clinical trialist, right? Someone who's doing the clinical research, and then we can pair with them to do some of the biology, and then they're bringing resources, and we write grants together to fund it all. And that's basically how it's worked. I mean, we have samples from at least 35 clinical trials and growing, and we've managed to I think most of the time satisfy everybody that we're returning results or returning research research data back to the projects in a reasonable time frame, with a lot of insight, with exciting results that are leading to the next thing. We're kind of constantly trying to make sure we're balancing our capacity with our efficiency, the projects we take on with our ability to deliver results. But so far we've been able to grow and grow, and it's been super fun. And as a result, I think, my lab has had access over the years to probably close to 10,000 blood samples now, which is just like an extraordinary number when you think about how rare the diseases are that we study. And it's really, I think, a testament to the people on my team that they're so collaborative, that they, are meeting with everybody. They're making, they're getting results back. So people feel like if they're collaborating with us, that, we're pulling our load and getting results back. And I think it also helps that there's a lot of patient and advocate enthusiasm for this technology, and people want to see this be available to themselves or their loved ones. And so I think that that's also motivating to the field to work with us.
No, that makes huge sense. And to that end, we at SARC, have collaborators as well. And, part of the other organization advocacy groups that we work with you as well. So I think it's, like you said, a win-win, and enthusiasm, the expertise that you can provide is huge. There's some other components of the infrastructure, and I don't know how much you are involved with this or how much you are comfortable in sharing, but the Osteosarcoma Explorer is a thing. Maybe you can share with the group, because we're jumping out of Ewing's, now we're going to osteo, and then we'll get back to the Ewing. So for those that are interested, what is that about, and explain a little bit more.
Yeah, well, I can't really take credit for the Osteosarcoma Explorer at all, but I can tell you a little bit about my involvement in it as sort of like like an enabler. I think is probably the biggest label I give myself in that space. But this really came out of a recognition from really the Quad W Foundation, which is a really important advocacy and funding foundation that think about a couple different diseases, but really, really focused on osteosarcoma. Their Quad W stands for what Willie want, and Willie Tishner was a patient who had osteosarcoma, and his parents are huge advocates in the osteosarcoma space and fund a lot of research through both Quad W and the Osteosarcoma Institute. And the Quad W organization recognized that there was, first they invested heavily in fixing the osteosarcoma samples that were banked at Children's Oncology Group, making sure they were better annotated so people could access them for research. That led to the NCI running a sequencing project on osteosarcoma called as part of the Target Project. And so that was a huge effort to sequence, I think it was a couple hundred, don't quote me exactly, a couple hundred osteosarcoma samples. And all that sequencing data is publicly available through the NCI's dbGaP, which is a way that researchers can get access to raw sequencing data from sequencing projects. And that would not have been possible had the Quad W not gotten involved with helping to better annotate the osteosarcoma samples that were in the bank at the Children's Oncology Group. But then there became, the next phase of that was a recognition that there were multiple projects happening potentially on the same tumor samples, often linked to those samples that were profiled by that NCI Target Project. And that it's hard for investigators who are not like my lab, who don't have computational biologists to sort of navigate that data and generate a hypothesis that's worth testing. And so the Quad W then helped partially fund uh, an effort by Dr. Yang Jie at UT Southwestern to develop this Explorer. And she had plenty of experience doing that in lung cancer before. And what it does is it really shows multiple different types of data that may have been done on similar, the same samples, and linked to some of the clinical data available from COG, so that people with more of a clinical research background can sort of explore the data visually and sort of generate a hypothesis that then they can test either through a clinical trial or through additional experiments. And, there's a couple of different ways to look at complex data from rare cancers, including osteosarcoma. But I think this is a tool that's really was developed specifically for osteosarcoma, specifically funded and envisioned by osteosarcoma advocacy group, and then really sort of executed by an expert group of people who know how to make these types of visual tools. And so, I think the more of that can happen across rare cancers, and the more we can pull together data from different research projects that are done across the country in different labs, the more discoveries that can be made by people who maybe don't have all of the sort of the math geeks that I have access to to help them analyze the data. And I think I think sometimes people hear and get the sense that the term data gets siloed, meaning it's stuck in one place, or that people aren't necessarily collaborating with their data. I mean, there's a teeny bit of truth to that, I think, raw truth to that in research. Like, everybody kind of has to publish their work to sort of make sure they can keep getting funding and secure their research programs and move forward in their careers. But honestly, the biggest hurdle, and the reason people sometimes keep their data to themselves, is that it's really hard and complicated to share data. It's huge. It's hard to transmit. You can't just transmit it without annotating it, without labeling it, without explaining what it means. And so, and then to stand up something that allows anybody to just explore it, even if they don't have expertise, like, it requires dedicated personnel. And I think people underestimate that. So what's so amazing about a group like the one that developed the Osteosarcoma Explorer is that's what they're good at. So now there's this place where people can and will continue to share complex data without having to also take on in their own laboratories this responsibility of visualizing it and making it intuitive to explore, which, to be honest, individual groups, even my own included, we don't really have the resources to develop those visualization tools. So it's a really important landmark moment for osteosarcoma, but also I think for rare cancer research in general, to have something like that.
Yeah, and that's exactly what we were thinking about. Quad W, as well as the Ticheners. Tichenor used to be the CEO of our board of SARC as well, and they're a great organization, as you said, but they had the vision that these patients are going on study, their samples are used all over the place, but it's the same sample, and how do you pull it all together? And there's a data for common good, as you are aware as well, and that's the same kind of mentality as to say this is the same sample, guys. We want to identify it, share it, annotate it in a standardized fashion so we can all use it, because in rare tumor space, we don't get that many samples, as you said. So I do like it. I appreciate you sharing whatever component you had to it. Thank you, because it's an important aspect to move forward. But you said the components, because people that are listening to the podcast might say, well, I have a very unique tumor group as well. How do I redo this for clear cell sarcoma, or let's say epithelial, what other tumor that we might want to have? And I think this is a way to get the collaboration moving forward.
Yeah, I do think it's a formula that can be followed, but requires, I think, some funding, advocacy, existing data, and someone with that expertise to really put it into like a visually useful exploration tool, which is not trivial for sure.
No, that's a huge lift, and it sounds really easy, but it took a lot of effort.
Yeah, that's right.
And then as we then take it from osteogenic sarcoma, I know you've been involved with consensus paper writing on the use of molecular characterization of Ewing sarcoma. And in the past, when we were trained, you didn't even need to do anything. It's Ewing's, it's CD90 positive. Okay, you just treat them all the same. But the reality, no, you have set some standards, and maybe you can share with our group how all that worked and how important that is to not just say you have Ewing's, but to be molecularly characterized. Because the same thing for rhabdomyosarcoma and any other rare tumor, we're starting to learn a lot more.
Yeah, well, I mean, to me, what was so striking when I was training as a oncology fellow was that that period of time was really when pediatric leukemia risk stratification based on molecular features and response to therapy was really taking off. And I mean, I remember people saying at the time, well, yeah, but we have these set of drugs and we give everybody these set of drugs. So will it really matter if we find out that someone's like resistant, like early on in therapy? Like, is that really going to make a difference? And, like it took some guts to say, well, let's find out the right way and not just assume we know the answer to that. And so, people developed molecular risk stratification schemas where the mutations helped predict how likely you were to be cured by standard of therapy, and then measured what we call minimal residual disease, which means teeny tiny amounts of leukemia still left in the bone marrow that you can't even see by eye when the pathologists look. And those tests really identified patients who almost for sure were going to relapse if they only got standard therapy. And only then, and only in that exact population, did adding three additional intense cycles of chemotherapy during that period after you found out that information, were they able to ask the question, would that help? Because they finally could identify that group. And it absolutely helped, right? That now patients with those like high risk features now have outcomes that look, when you intensify therapy, have outcomes that look just like patients who have standard risk pediatric leukemia, which is like over 90% survival now, which is like amazing. So I think we're just at that phase where we're primed and prepared to identify the group, not based on clinical features alone, but really on genomic features, with clinical features as being a part of it, and response to therapy features that really needs for sure that intensification of therapy. And then I think we have to ask the question in a clinical trial, would additional intensification or some change of therapy early on make a difference? And we shouldn't assume that since we've tried intensification for patients that we identified halfway or longer down into therapy, it didn't make a difference, that if we could apply it earlier, that it wouldn't make a difference. That hypothesis has not been tested and needs to be tested. I think the other piece that people may underappreciate, because a lot of us think about improving survivorship so that 100% of our patients survive. We know that the current intensity that we consider standard of care for Ewing sarcoma hasn't been the intensity that we've always used. We know that less intense therapy didn't cure as many patients, but still cured like 50% of patients with Ewing sarcoma when the first successful chemotherapy was started, right? So that means we're now over-treating some group of patients, and that comes at a cost. That comes with significant chemotherapy-related and treatment-related, often lifelong toxicities that people have to live with, and in the case of pediatrics, ideally for 50, 60, 70 years. So I think there's also an opportunity that to identify patients who are extremely likely to respond to even less than we'd consider standard of care now and still be cured and decrease those toxicities. If we could identify them carefully, and we're going to definitely need molecular features and response to therapy early on, response to therapy features to be sure that we're selecting only the right patients for dose reduction, because we don't want to reduce and then have someone not survive. So we only really want to reduce therapy in patients who are going to survive, be cured by that reduction, and then also take away some of the long-term toxicities that we know our patients have to deal with. So I think there's like an incredible opportunity. I mean, I'm sure you can tell in my voice and my face that I feel really passionate about this. Like this is the the pie piece that I think my lab is going to have a part in contributing to, is improving our understanding of risk stratification, improving how we're improving the precision with which we intensify therapy or de-intensify therapy, and that will contribute to improving outcomes. On the other hand, we still need people out there who are making discoveries on new therapeutics and understanding basic biology and early detection of cancer development and cancer risk syndromes and all that stuff. So, I mean, it really is going to take a community for us to do better, but this is our piece of the pie. This is our little contribution to the big picture, I think.
So I didn't use the word little. I think you used the word slice of pie. This is a big component because this is a huge step forward for all of us. And, a lot of us that do sarcoma work, we think we need to treat more and more people. But what you're really seeing, the better outcomes is actually stopping therapy when they don't need it anymore. So I do think this is maybe something that a pediatric, we should do it in adult world too, is only treat people that need to be treated and stop so they don't suffer the side effects, and or it looks like they're not responding, don't keep going the same before imaging and then do a switch and pivot. So I do think that this is a model going forward for Ewing sarcoma. But we're a rare tumors. There's 100 different tumors. So how many more labs are we going to have for you? We're going to need every single lab space that you have for this as we move forward. So, but I do like this. And as we're getting close to the end here, I'd like to kind of have our audience hear a little bit of what your hope and vision and goal for this type of space, not just in Ewing's. And you kind of shared it already, but where do you see us in your hope and goals that we go with this? Because you've done it for Ewing's, you're doing it for others as well, but where do you see that going?
Yeah, I mean, we've talked a lot. I think we've mentioned most of pieces of the vision as we've gone, but I think ideally patients who are who have a new lump who come in, a part of their diagnostic workup is getting a liquid biopsy test. I actually don't think it's crazy to think that 10 to 15 years from now, you have a liquid biopsy test sent off at the same time the initial diagnostic biopsy gets scheduled, and then maybe you cancel that because the liquid biopsy tells you convincingly what they have. And if it doesn't, okay, then you get the biopsy anyway and you figure out definitively what they have. And then also the molecular features you detect from that liquid biopsy, and either alone or together with the biopsy itself, help you predict, help you choose the intensity of your therapy. And then you measure that response really maybe within 2 weeks, 3 weeks, 4 weeks of starting therapy, so that you can adjust the intensity, or even in some cases, I think you were sort of alluding to this, maybe pivot on to something that is experimental early rather than waiting until resistance disease has developed, because you already know for sure that the standard isn't going to be the right option for that patient. So, and then, for patients who get, good response to therapy and have no evidence of disease on their imaging, which is always our goal for every patient, of course, I think this additional test, probably not the same test, but more sensitive, maybe even patient-specific tests will be used as part of the off therapy or even on therapy surveillance strategy to make sure that if the disease does come back, that we catch it early. And then again, to monitor as we go the effectiveness of any therapy that's used in the recurrent setting. I think you could imagine that if you catch recurrences early enough that they're probably still going to be harder to cure, but could you create more of a chronic disease scenario similar to how HIV is treated these days? It's extremely unusual for a patient to pass away from HIV, but we don't cure it. We just keep it at bay all the time. And so I think, but the only reason that works is because there are blood tests that tell their clinicians whether the antivirals they're getting are effective at suppressing the viral load. So I would love, 20, 30 years from now, if we're doing something where, perhaps perhaps patients have had their first relapse and cure maybe at that time still off the table, but we can keep it at bay for 5, 10, 30, 40, 50 years until the next cure does come around, right? I mean, that would be a whole game-changing thing. So I think that there's also opportunity to think, this is years down the road, but to think about this as a way to also turn cancer into a symptom-free chronic disease would be amazing as well. And then the last piece is that in my outpatient clinic, I do pediatric cancer risk syndromes. So we're also really interested in adding liquid biopsy to our surveillance for patients we know have a genetically inherited risk of developing cancer but haven't yet. We're always sort of trying to make sure we catch if they develop cancer, we catch it very early. And I think liquid biopsies could be one more tool in our toolbox for early detection. And then we already know that if we detect things early, we're much more likely to cure patients. So I think that could also have a big impact. So that's sort of like the 5 to 20 year vision, I think, for how we think liquid biopsies are going to have an impact. That doesn't. The one last thing is I think we're going to learn scientifically a whole bunch about how these tumors evolve and change over time, because it's so easy to sample the tumor now from a blood sample at many times during a patient's treatment course and their cancer journey. And so scientifically, I also really hope that we're going to give insight into tumor evolution in a way that no one's ever been able to uncover before in some of these rarer cancers.
Well, for those that are listening, this is a lot of great information and visioning that goes along. And I think all of us that are listening to this, say the future is liquid biopsy, partly because it's accessible. The hematologists have had access to this forever. Where the solid carcinomas have had access to it. We're slowly working into the rare sarcoma space in this, because repeated biopsies are very difficult in our space. So having a liquid space is huge. And we looked at Dr. Crompton and his lab and other collaborators to really move this along and incorporate it into our practice. So we do appreciate Dr. Crompton for sharing your time with us. And we hope that your work continues in this space, and your enthusiasm and knowledge is infectious and goes to a lot of different collaborators. Is there a way people can get a hold of you that we can put on our notes?
Sure. I mean, my email, I think, is Googleable. And we have the website where you can actually, our website, it's cromptonlab.org, is also has an email space. But we're not hard to find. We try to go to a lot of the meetings. If you're at one of the sarcoma related meetings or ACR or ASCO, feel free to grab me. I love to talk about this stuff, obviously. So I'm always happy to chat with people from whatever spectrum, whether it be, sort of advocates, industry, family members, other scientists, clinical researchers. There's always lots to talk about. It's a really exciting space, and I'm thankful to SARC for giving me an opportunity to share it with the community in more detail.
No, we thank you for the opportunity, and it's Crompton Labs, and that is on the web, and they have opportunities to help support his lab as well. So we look forward to that. So again, thank all of our listeners here for another SARC Talk, and we look forward to our next SARC Talk, and hopefully everybody has a great rest of the summer. Thank you.
Thank you for listening. Be sure to subscribe so you never miss an episode of SARC Talk. To find out more about SARC, please visit our website at sarctrials.org. To suggest a topic or for questions, please email us at sarctalk@sarctrials.org. The SARC Talk series is made possible due to funding from Boehringer Ingelheim. Together, we can find a cure for all forms of sarcoma.